Hello everyone, welcome to my channel Bani's Biology. Today we will learn about the elongation and termination process in prokaryotic DNA replication. I have already discussed about the detail of initiation process. If you want to know about it, please go and visit. The link is in the description below. Before starting the elongation process, let's quick recap the initiation process. DNA replication start at a specific site, that is ORI site. This is the ORI site. An initiator protein like DNA A protein that bind to ORI site and then helicase bind this ORI site and helicase unwind the DNA and primase lay down the RNA primer. This set the stage for the elongation process. Elongation process proceeds in bi-direction. This is one direction, this is the another direction. This is the origin of replication site. In elongation process, DNA polymerase 3 is the main enzyme responsible for synthesizing new DNA. It adds nucleotide to the growing strand. This is the growing strand and the 5' to 3' direction using the parent strand as a template. DNA polymerase 3 require free 3' OH. Without 3' OH, DNA poly 3 cannot synthesize the new strand and cannot add nucleotide, and primer give the 3' OH site. This is the DNA polymerase 3 holoenzyme. In holoenzyme present beta clamp and core polymerase and gamma complex. This gamma complex is the sliding clamp loader and beta clamp is the sliding clamp. DNA polymerase 3 has high processivity, meaning it can synthesize thousands of bases without falling off. It also has probreeding ability via 3' to 5' exonuclease activity to correct mismatches. DNA poly 3 alone is not stable on the DNA. It tends to fall off after synthesizing a short stage. To solve this, the beta clamp, that is sliding clamp, is recruited. Beta clamp is a ring sepid. This is the ring sepid that encircle the DNA and hold DNA poly 3 in place and increasing its processivity. After adding RNA primer on the ORI site, clamp loader complex, that is gamma complex, recognize the primer and template junction, and clamp loader that uses the ATP to open the beta clamp. This is the process how the ATP help in the loading the sliding clamp. This is the sliding clamp loader and that help in the loading the beta clamp in DNA, and sliding clamp loader uses ATP as an energy source to open the beta clamp and load it into the DNA in the site of primer and template junction, and that is recognized by the sliding clamp loader. And once the clamp is loaded, DNA poly 3 binds to the clamp. This forms a stable DNA poly 3 clamp DNA complex, allowing continuous synthesis. The clamp act like a seat belt, securing DNA poly 3 to the DNA for high speed processive synthesis. Once the clamp is loaded, DNA polymerase 3 recruited to begin elongation process. This is the elongation process. In DNA, one template strand is 3' to 5' and another strand is 5' to 3'. In 3' to 5' synthesis, allowing DNA poly 3 to synthesize in the 5' to 3' direction and matching the fork movement. This is the fork movement site that matches the fork movement site. It requires one primer, and this strand is called leading strand, and this strand synthesize continuously without breaking, and the clamp is loaded once at the origin. This process is smooth and efficient. On the other side, template strand is 5' to 3' direction, but DNA is synthesized to 5' to 3' direction. It synthesizes opposite side of fork movement. This is the opposite side of fork movement. It works away from the fork. This result, DNA poly 3 synthesized short fragment called Okazaki fragment, and scientist Raizi Okazaki discovered this fragment. That's why this fragment is also called Okazaki fragment. Each fragment begin with new primer. This is the primer and this is the primer. So multiple primer are required. The clamp must be reloaded for each Okazaki fragment. This strand is synthesized discontinuously. This strand is called lagging strand. This is the picture where you can easily understand how the lagging strand is synthesized. This is the ORI site, and here is the two direction of fork movement. This is the one direction fork movement. This is the another fork movement. If I focus this one side of fork movement, you will see this picture. One is 3' to 5' direction, another is 5' to 3' direction, and here helicase continuously unwind the DNA by breaking the hydrogen bond. This strand is leading strand, that is 3' to 5', and this strand synthesized continuously, and another strand synthesized discontinuously. This strand is called lagging strand, and this is the first primer. And the first primer is added, is continuously DNA synthesized 5' to 3' direction up to end. This is the end position. The DNA is continuously synthesized. Next, helicase continuously unwind, and as replication proceeds, another primase came and synthesized the primer. Then another fragment will synthesize on the lagging strand. So this is the second primer. Same way as replication further proceeds and continuously leading strand will synthesize continuously, and lagging strand makes small fragment like this. This way lagging strand is synthesized. To match the speed of leading strand, scientists proposed a model that is called Trombone model. In Trombone model is a conceptual framework that explains how the replisome synthesizes both DNA strands simultaneously. In Trombone model describe how DNA polymerase 3 coordinates leading and lagging strand synthesis at the replication fork by looping the lagging strand. This is the loop formation, allowing both strands to be synthesized simultaneously. Its name for the way the lagging strand loop out and collapses repeatedly, resembling the slide of a trombone. In this particular model, scientists have proposed that the DNA polymerase holoenzyme have 3 polymerase. For leading strand, needed 1 polymerase for sufficient continuous synthesis. This is the leading strand. Only 1 polymerase is needed. But lagging strand need 2 polymerase to alternate between Okazaki fragment. This is the 2 polymerase. This is the lagging strand for improving efficiency and coordination. This is the full elongation process where is the primase is added, and then DNA polymerase 3 holoenzyme continuously added and synthesize the leading and lagging strand. And this is the loop formation of lagging strand. Once the DNA polymerase 3 finishes synthesizing the fragment, the RNA primer must be removed. So RNA primer removed by the DNA polymerase 1. First, DNA and RNA hybrid is removed by the RNase that only degrades hybrid condition except the ribonucleotide. This is the ribonucleotide site directly linked to the DNA end that is removed by the 5' exonuclease, and the DNA polymerase 1 fill this gap until every nucleotide is base pair. But this nick is repaired by the enzyme DNA ligase with high energy cofactor ATP that create a phosphodiester bond between adjacent 5' phosphate and 3' OH. With this, elongation phase reaches completion and setting the stage for the termination. This is the structure of the DNA polymerase 1 that have 5' to 3' exonuclease activity and 3' to 5' exonuclease and polymerization activity. And this is the large fragment that's called Lenau fragment, and this is the small fragment. So DNA synthesized bidirectionally. Once the replication fork have moved around the circular chromosome and DNA synthesis is nearly completed, the process must stop. The termination region contains a specific TUS site, and the protein called TUS, termination utilization substances, bind to this TUS site. There are 2 phases of TUS and TER complex. One is permissive, another is non-permissive. This is the structure. One is the permissive phase, another is the non-permissive phase. When the replication fork encounter the permissive phase, the helicase can displace TUS and proceeds. This is the permissive phase, and the DNA continuously unwind and synthesis proceeds. When the fork hits the non-permissive phase time, separation trigger a lock conformation of the TUS and TER complex. Holder further unwind. The completed chromosome joined as a catenate or topologically interlinking circles. The circles are not covalently linked, but they are interwound. This catenated chromosome separated by the type II topoisomerase, that also called DNA topoisomerase 4, transiently breaking both DNA strand of one chromosome and allowing the other chromosome to pass to the break.